Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Risk-Based Monitoring in Clinical Trials, Improve Oversight Through Intelligent Review of Digital Endpoint Data. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, which is found on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions to the clinical trials industry, generating high-quality clinical evidence for life sciences companies. They offer comprehensive evidence generation solutions that combine medical imaging, eCOA, precision motion, cardiac, and respiratory endpoints. For more than 50 years, Clario has delivered deep scientific expertise and broad endpoint technologies to help transform lives around the world. Their endpoint data solutions have been deployed over 30,000 times to support clinical trials in more than 100 countries. Their global team of science, technology, and operational experts have supported over 70% of all FDA drug approvals since 2012. Now it's my pleasure to introduce and welcome our speakers for today's event. And first, I'd like to welcome my co-moderator for today's event, and he is Dr. Todd Rideau. Hello there, Todd. He is the Chief Medical Officer at Clario, where he is leading medical and scientific strategy across the organization. Dr. Rideau brings over 20 years of experience in pharmaceutical research and clinical cardiology, supporting customers with scientific and regulatory strategies for their development programs. Next, I'd like to welcome and introduce you to Dr. Stacy Brummerman. She serves as VP of Clinical Science for Clario's eCOA Neuroscience team. Hello there, Stacy. Leading strategic clinical and scientific oversight of eCOA assessments and scientific services across global psychiatry and neurology programs. She brings over 20 years of clinical experience and more than a decade in CNS-focused clinical research. Next, I'd like to welcome Kevin McCarthy. He's a Senior Principal Scientific Affairs Advisor, Respiratory Science at Clario. Hi, Kevin. And Kevin has worked in lung function testing for 53 years, with 40 years as the manager of the Pulmonary Function Laboratories at the Cleveland Clinic Health System. He was on the task forces that updated the ATS-ERS standards for spirometry and lung volume measurements. And next, I'd like to welcome Dr. Alon Gay, Vice President of Scientific and Medical Affairs, Cardiac Solutions at Clario. Hi, Dr. Gay. He is a board-certified cardiologist with experience in clinical practice, cardiac electrophysiology, and 15 years in drug development and medical affairs. At Clario, he contributes to the ECG Core Lab, interprets Holters and other cardiac recordings, and he also consults for clients around the world. So that is Alon. So thank you very much for everyone joining us here today. And with the introductions out of the way, it is now my pleasure to pass the mic over to Todd. So Todd, when you're ready, you may begin.
Okay, great. Thank you, Sonia, for the very kind introductions. Thank you to all of you, our quite sizable audience that has joined us today as well. It's great to be back for another monthly educational webinar today. I'm particularly excited about this one as there's quite a bit we have packed into the session today. Focusing on novel approaches to risk-based monitoring. What you're going to hear today is a bit different than the more commonly applied risk-based monitoring or risk-based quality monitoring methods, as we're talking about more specific approaches to particular types of digital endpoint data. So we'll take you through this agenda today, starting off first with just a brief overview of what is risk-based monitoring. Following this, Stacy is going to take us through how the RBM principles can be best applied to ECOA data, and we'll walk us through a few specific use cases and examples there. Kevin is then going to jump in and spend some time focused on spirometry data and best practices around RBM there. Spirometry is a very unique data type, and as you'll see, requires a unique approach for proper oversight. And then in the last segment, we'll hear from Dr. Gay, taking us through several especially innovative tools for monitoring cardiac data, so specifically for continuous ECG, standard 10-second ECG, and blood pressure as well. And then at the end, I'll wrap us up and lead us through a Q&A session. Please do send in questions as we go. We'll do our best to get to them, and anything we don't get to, we'll certainly follow up directly by email. So just to get an understanding of this, you can go to the next slide, please. Just to get an understanding of this audience's perspective, we asked you all by survey during registration and on LinkedIn, which area or data type presents the greatest challenge for risk monitoring in trials. So here are the results of the survey, and thank you all for your responses. The four choices were general site level performance and monitoring, ensuring ECOA data quality and compliance, oversight of spirometry data, and then the assessment of cardiac safety data quality. And as you see here in the results, the hottest area of interest is in general site performance and monitoring. And that is important, and probably what most of you, this audience, has been exposed to in your work in drug development in the past. Following this was ECOA data quality and compliance. This is a specific data type and has broad importance across a range of different therapy areas and indications. Respiratory and cardiac followed with a smaller percentage of you considering it the greatest challenge, at least. I think you'll be pleasantly surprised, though, to learn about how problematic both spirometry and cardiac data types can be in terms of ensuring data integrity, that is. And it'll be good to learn about what we can do here to keep a study on track. So next slide. With that overview of what we intend to cover today, I think it's important that we start here to level set us all on what we mean by risk-based monitoring. So at a high level, RBM is a method for effectively and efficiently monitoring and managing data quality throughout the duration of a clinical trial. The objective is to ensure the accuracy and reliability of data, but without implementing broad and systematic measures that are deployed uniformly across all sites in the same way, and therefore introduce a very high resource burden. The idea is to develop an approach to identify signals for potential risk and then increase the level of monitoring in a focused manner around those risk signals. So the goals overall, number one, safeguarding participants, ensuring sites are collecting the right data in the right way, that there's no emerging safety signals that could be missed and need to be addressed. Ensuring compliance with regulatory expectations for study monitoring, obviously a second objective, more about that on the next slide. And last, efficiency and prioritization. So ensuring the efforts and resources are spent where they will be most impactful. You can go to the next slide. So I mentioned meeting regulatory expectations as one of the key objectives. Here on this slide, I've called out just a few kind of key regulatory milestones, if you will, to be aware of. This is something that's evolved over quite some time now, originating with two guidances back in 2013. One each from FDA and EMA, urging the industry to adopt a risk-based approach. This opened the door to pivot from current practice at that time, which was 100% site monitoring every 4 to 8 weeks on site, and then evolving now to, or at that time, to something more centralized, leveraging technologies, and specifically calling out that this approach would still meet the requirements for site monitoring. So this was progress, very pragmatic and thoughtful approach, now endorsed and could subsequently evolve further. In 2016, it was the second revision of the ICH E6 guidance that spoke to flexible approaches and emphasizing the role for central monitoring to complement or augment on-site monitoring. And then some years later, the MHRA guidance followed, similarly emphasizing a thoughtful and technology-enabled centralized approach. Next slide. My last slide here, just to recap what we're going to be talking about or why we're talking about this today, the key benefits of a well-designed and well-executed risk monitoring plan. So again, participants, from a safety perspective, you have a drug without a defined safety profile, and the accuracy and timeliness of safety data, therefore, is paramount. An effective RBM program is critical for ensuring that sponsors have the data needed to make the safety-focused decisions during conduct. Second, data integrity and reliability. Again, not just about safety, it's also about efficacy determination. So small sample sizes, limited power means that making every participant count is essential. Poor quality data means an inability to detect signal within noise, and therefore go, no-go decisions can't be made confidently. So monitoring site performance includes specifically looking at the digital endpoint data that informs primary and secondary objectives, which is why you'll hear much more about this today. Third, costs, so reducing unnecessary monitoring expenses by expending resources in an efficient manner, a key benefit over the traditional practices, certainly. And then last, regulatory expectations again. So we're going to be digging into specific data types that sites collect, talking more about some tools that are a bit distinct from general site performance metrics that are more typically tracked, and how they provide a unique risk-based perspective. Next slide. So to take it from here, I'm going to turn over to Stacy Rummerman, who's going to be talking for about the next 15 minutes or so, speaking about what a good percentage of you identified as a key aspect of RBM, and that is the application to COA data. Stacy?
Thanks, Todd. Good morning, everyone, or good afternoon for those of you outside of the U.S. Pleasure to be here today. My name is Stacy Rummerman, and I oversee our ECOA Neuroscience Clinical Science team. Now, what I'm going to talk about today is ECOA as risk-based monitoring as it pertains to ECOA. Now, my team specifically manages more neuroscience studies, so we're teams of clinical neuropsychologists and SMEs who help oversee the training, clinical, and data oversight of trials. But what I'm going to speak on today is not specific to neuroscience. There's certainly many applications well outside of neuroscience. And I was glad to see the interest in ECOA, not surprising. One of the things that we're seeing is that ECOA platforms are increasingly used to capture primary and key secondary endpoints across a wide range of therapeutic areas. This means that the data collected directly from participants, caregivers, or site clinicians has a direct impact on study outcomes. These endpoints often include subjective ratings, symptom severity scales, patient-reported outcomes, and performance-based tasks. From a risk perspective, this type of data is sensitive to rater effects, learning curves, and deviations in timing and administration. Data is also generated in high volume, often in near real time. Together, these characteristics make ECOA data both critical and complex, and highly relevant for structured risk-based oversight. Traditional periodic monitoring approaches are no longer always sufficient to keep pace with the volume and the velocity of the data coming in. At the same time, the industry is shifting more towards data-driven, risk-based monitoring models. In this context, integrating ECOA with risk-based monitoring allows us to proactively identify and manage risks, rather than reacting after issues have occurred or after endpoints have already been compromised. The ultimate goal is to protect data quality by identifying potential concerns earlier and more efficiently before endpoint integrity is at risk. Next slide, please. So first, I want to define what ECOA is. It sounds like this audience has a pretty good understanding, but I recognize that not everybody speaks in acronyms all day. So ECOA is electronic clinical outcome assessments. So it's the electronic capture of COA data. So your formerly paper-based assessments, your PROs, your clinician-reported outcomes, all have been moved into validated digital platforms for administration and scoring. That's what ECOA refers to. There are certainly some studies that are not using platforms, but more and more we're seeing reliance on these platforms. And often our ECOA measures are the primary or key secondary endpoints and can include both subjective and performance-based measures. This is especially true within the neuroscience space, but not entirely unique to neuroscience. These assessments can be rather complex, prone to rater drift, and challenging to standardize. for a dementia trial looking at cognitive impairment, for example, or a depression study relying on clinician-rated symptom severity for depression. The standardization of both administration and scoring of the selected ECOA measures is critical to the success and the outcome of the trial. Now, what's great about ECOA is that we now have real-time access to data, allowing for quicker oversight and monitoring. Many ECOA platforms also have built-in logic such as edit checks or flags for missing data that improve data quality right from the point of data capture. This allows us to identify variability in administration early and engage with sites to ensure ongoing calibration. It can be a first basic tool to start and support your risk-based monitoring plan. ECOA platforms also support regulatory requirements through features such as timestamping, audit trails, and have the ability to greatly improve protocol compliance. Next slide, please. Now, there's a number of risks that may be identified or concerns to consider when using ECOA, and the data associated with these risks should be considered on multiple levels. So starting with our participant level, we have better visibility into participant level signals such as missed data, unusual response patterns, inconsistent reporting between patients or participants and their caregivers, and even placebo response, which we know can greatly influence study outcomes. We can also identify concerns on the site level, such as frequent rater changes, scoring anomalies, site rater bias, and unusual ECOA completion times. Data may also flag on the study level, suggesting concerns with visit schedules themselves or even training quality. The amount of data collected across participants, across measures, and across sites allows for a high volume of data, all in one place, but also necessitates a monitoring approach which is timely, specific, and efficient. Next slide, please. So traditional monitoring approaches can now be replaced with more targeted risk-based monitoring approaches, allowing for early identification of data issues, especially when the approach is customized to the study. With the volume of data available, risk-based approaches allow us to focus first on the critical ECOA endpoints and key time points defined by each protocol. From there, we can prioritize which signals or risks require action and identify sites or raters that might benefit from additional oversight or support. Using analytic-based approaches allows us to also detect trends or patterns over time and ensure we have continuous oversight of the data coming in, enabling timely intervention when needed. Next slide, please. So there's a number of approaches that monitoring teams and sponsors can employ to support risk-based monitoring and ECOA, risk-based monitoring of ECOA, I should say. With advanced data analytic approaches, we can combine broad data surveillance with targeted signal detection. This approach brings together descriptive, longitudinal, and comparative analyses at multiple levels: participant, site or rater, country, or even study level. Some common methods include identifying response patterns and anomalies, assessing variability and drift at the site or rater level, or even evaluating whether assessments are completed on time and in line with protocol requirements. We can also apply algorithmic-based approaches that rapidly flag potential data issues. These can include item level or assessment level errors, as well as inconsistencies observed across visits or between ECOA measures. The objective here is to transform raw data into structured, actionable signals that can be incorporated into your risk-based monitoring reviews. This enables study teams to determine when further investigation, retraining, or other interventions may be needed. Overall, the focus is on maintaining the quality, consistency, and integrity of endpoint data, allowing teams to act quickly and appropriately. This structured approach ensures that risk-based monitoring activities are they're scalable, they're precise, and they're actionable. Next slide, please. Analytical methods can identify patterns that suggest an assessment may be atypical and flag specific data points that require closer review. This is where independent review can add value. Independent review is a more in-depth evaluation of source data, often including audio or video recordings alongside review of the source. It leverages clinical experts to focus on a targeted subset of assessments, often but not always those identified as high risk or atypical, and evaluates adherence to administration and scoring standards, as well as the rater training. The primary objective of independent review is to identify potential administration or scoring issues and to provide feedback to sites to support retraining or recalibration efforts on an ongoing basis. While independent review can be more resource intensive to implement, it becomes highly efficient when paired with risk-based monitoring approaches such as analytics. Together, these methods allow us to focus review efforts on assessments with the highest likelihood of error. Now, one key benefit of pairing risk-based monitoring services with ECOA is access to large data sets that can be used to validate signals and refine algorithms, strengthening the risk-based approach. This combination is especially valuable when findings require deeper evaluation or interpretation and where clinical and instrument expertise are critical. Next slide. And this brings me to my last point. The design of risk indicators and thresholds for ECOA flagging, unfortunately, it cannot be entirely standardized quite yet. Risk indicators and thresholds depend on the property of the instruments used, the disease course, and expected variability of the target population. To do this effectively, teams supporting risk-based monitoring from design through implementation need deep expertise in both the population and the COA measures being used. This is critical to distinguishing normal clinical variation from signals that may indicate measurement error, rater drift, or bias. For example, even across two depression studies, the appropriate flags and action thresholds may differ. A trial evaluating a traditional slower-acting treatment will have different expectations for symptom change compared to a study using a rapid-acting compound. The investigational product, the visit schedule, frequency of COA administrations all need to be considered when defining your risk-based monitoring strategies, in addition to other protocol-specific factors. This reduces the risks of false positives or negatives in risk-based monitoring and ensures that any actions taken are meaningful and deserving of both the monitoring team's time and the time of the site. Ultimately, carefully designed, expert-informed methods strengthen both the scientific rigor and regulatory defensibility of efforts to ensure the digital endpoint quality. Next slide. In closing, as trials become more digital and data-rich, the ability to intelligently monitor the large number of data and critical endpoints becomes essential. Integrating ECOA into risk-based monitoring frameworks allows us to move from reactive to proactive oversight, ultimately strengthening trial quality and reliability. Doing so with the oversight of clinical experts to help guide your risk-based monitoring implementation from conception to the actioning of findings can further ensure data quality is maintained for the life of the study. With that, I thank you for your time, and I will now pass it over to our next presenter, Kevin.
Thank you, Stacy. So let's jump in here. As Todd mentioned, spirometry measurements are sort of inherently risky. And most people have, in the past, in clinical trials, the typical way of dealing with that is to have the measurements undergo an assessment of how acceptable the data was. And that's according to certain standards. International standards are published every several years. And that would help them establish whether it's acceptable, and then repeatability of repeat measurements, repeat efforts done within a measurement helps assign a letter grade, and that assigns a degree of confidence to the data, and that's typically where people sort of end that, thinking that this is going to ensure that my data is good quality. Next slide, please. But what we found in looking at data over the last several years, particularly since the latest set of standards were published, is that acceptable and repeatable spirometry may still be inaccurate. And a lot of our efforts have gone into trying to identify data that falls into that category to try and help our sponsors try to protect them from including that. So it's spirometry, if you look at the next slide, please, it's dependent upon, it's unique in that it depends upon the patient really understanding what it is you're asking them to do. It's a physical act, and it has to be done with maximal effort. And there are several points during that effort, the maneuver, that are really quite uncomfortable for the patient for a very brief period of time. What that means is it's not excessively uncomfortable overall, but patients wouldn't voluntarily take themselves to their maximum level of performance unless they were being coached to do so. And so the understanding of the measurement and what can go wrong, what can go right, is really important for the operator to have. That typically comes with experience. And it's the interaction between the operator, the patient, and even the collection device, which can provide the operator with certain clues as to how to improve the data between each effort, kind of make the next effort better than the one that just happened. Next slide. So as I've said, we've looked at a lot of data, and what we found that data that was rated acceptable and repeatable by overread can still show excessive variability. That's the noise that can sometimes hide the signal that the clinical trial is trying to uncover. And in respiratory clinical trials, the spirometry typically provides the primary or key secondary endpoints. And so it's really, really important that we go beyond just the standard, did it meet ATS acceptability, and look at things like physiologic implausibility, larger than expected changes between visits or after an intervention that was typically designed to produce a relatively modest change. But what we've uncovered is something that is a major contributor to the noise in spirometry in clinical trials. And it turns out it's a fundamental execution error, next slide please, that really isn't attributable to the patient themselves. So, I said the patient has to cooperate fully, and even if they are trying to cooperate fully, this type of error, called submaximal inhalation error, can only be attributed to the operator themselves. And it's actually quite understandable how it occurs, and I want to go into that and help you get a better understanding of all the factors that conspire to make this not uncommon. So it turns out this error is really, boils down to one thing. The command to start the forced exhalation is given before full inflation has been reached. And because it's at a submaximal lung volume, everything else about the forced exhalation that follows the deep inhalation can be perfect. And yet it's not data that you want in your clinical trials if it started far enough below full inflation. It can be difficult to recognize during testing. In fact, before the 2019 standards, it was impossible to recognize during usual testing. Next slide, please. So to give you a better understanding of what's happening here, this is what's called a volume time tracing, and it represents volume going in and out of the lung during a typical forced spirometry maneuver. On the left-hand side, we see those three little bumps. Those represent what's called tidal breathing. That's resting breathing that your respiratory center will initiate and maintain. You don't even have to think about that. But let's look at phase one of the test. That's where the operator coaches the patient to inspire as deeply as they can. So they really have to vigorously coach them to get that last 5 to 10% in. That's the part that I said the patient doesn't willingly want to do. When they do reach full inflation, we hit phase two. This is the start of the forced exhalation. This is where submaximal inhalation error comes into play. That it's not uncommon for the operator to really be a little bit uncertain about when full inflation is reached. Add to that the anxiety that if they hold them there too long, they have the knowledge that patients have a tendency to leak a little bit of air out before maximum effort is applied. So while they're waiting for the command to blow, a little bit of air is leaking out. And if too much air leaks out, then this all-important parameter, FEV1, which I'll explain in a second, can be rendered unacceptable. So it's that sweet spot of when to give the command to blow that is causing the problem here. The patient, once they start the exhalation, they continue exhaling with maximum force until they reach a point where they're either empty or close enough to empty to end that exhalation to keep the patient safe. And then the operator rapidly switches to coaching that has the patient breathe back in again as fast as they possibly can. And you notice that the total volume exhaled, FVC, that stands for forced vital capacity, and the total volume inhaled back again afterwards is the forced inspiratory vital capacity. In this slide, they match, and that represents exactly why this fourth phase was added in the 2019 spirometry standards. Next slide, please. So why is it so common? What we've seen is that up to 15 to 20% of measurements that look perfect otherwise demonstrate submaximal inhalation error. Well, in the standards that, as I said, were published every several years, it wasn't until the 2019 standards that they actually addressed how does the operator know when to give the command to blow. So specific guidance was given in the most recent standards. But for years, spirometry typically taught from one person to another. That wasn't, that was kind of like just whatever works. And we'll go into that in just a little bit later. It's a little bit difficult to judge exactly when that point has been reached. And it's almost impossible to do it with in a reliable way if the operator, while they're giving the command to breathe in deeply, if they're focused on the display of the spirometer, then they're missing what is the most valuable feedback that can inform them on the timing of that command to start. Next slide. And add to that, as I mentioned, that there's a start of test error that can cause the FEV1 to be considered unacceptable. And if that occurs, then the tendency is for operators to go, okay, well, I'll give the command a little earlier this time. Or they adopt a way of coaching that just says, okay, I'm going to say, take a deep breath and blow and hope they're at full inflation. And then what confounds that assessment of full inflation is that it used to be taught that if you demonstrated repeatability in at least two efforts within the measurement, repeatability of the largest FVC and FEV1, that meant that they were at full inflation. That's a falsehood. We now know that's not true. And all of these things conspired to sort of reassure the operator that they were having a good technique in play, when in fact the 2019 standards with the addition of that phase 4 now informs them that they may not. Next slide. So what we did was we looked at a large set of data, pooled data from 3 different asthma studies, and focused in on the measurements that were judged to be acceptable and demonstrated repeatability. And those 15% of all the measurements in those 3 trials did just that, but also showed SIE, submaximal inhalation error. On average, they started from about 1/3 of a liter below full inflation. And that may not sound like much, but in the context of asthma and COPD drugs, that can equal or exceed the signal that you're trying to tease out in terms of looking at significant changes. And in every one of those studies, there were outliers that were greater than 1 liter below full inflation. And that can really confound the data analysis if that gets into your study set. Next slide. So FIVC, we won't go into the math here, but it's simply a comparison of what they breathe in afterwards to what they just exhaled. And if that volume, the difference between those 2 volumes exceeds 5%, that confirms submaximal inhalation error. The emphasis on this slide is that we now finally have an objective means to evaluate full inflation, and we never did before. And you would think that would sort of clean up everything right away. Next slide. Unfortunately, it doesn't. And so what we're experiencing now is that, the FIVC was actually designed to be a tool that the operator could use to evaluate how his timing was, how well he hit the mark. And if submaximal inhalation error was indicated, they could go, okay, I'll try to hold off and give that command a little lower. That's the appropriate way to use the FIVC. The inappropriate way to do it is to, when it flags a submaximal inhalation error on one effort, on the next effort, have the patient inhale that inspiratory vital capacity more slowly, giving the operator time to end it when it matches. And that's what we're seeing. Next slide, please. So it's data that's not good, that's made to look good, and certainly that was never the intention of the standard. Looking at the upper left there, we see FIVC doing exactly what it was designed to do. It recognizes that there's about a third of a liter difference between what was inhaled afterwards, and that tells us the forced vital capacity started too soon. Down below is another example of submaximal inhalation error objectively demonstrated, and it bears some explanation. What you can see is that sharp peak there at the start of the forced exhalation. That peak matches the volume of the two small tidal breaths that preceded it. In other words, the very first requirement about spirometry, that it start from after a maximal inhalation, that was never achieved. We don't need phase 4 to show it, and yet we do have a phase 4. And it suggests that really did start from full inflation, and that can occur. This is the kind of thing that we're seeing. And so submaximal inhalation error is definitely present in the lower panel, just wasn't detected by the FIVC. That's a little disappointing, but that's the reality. And that's one of the things we want to alert sponsors to, is the sites that do that kind of testing need to adjust their technique for the FIVC. Next slide, please. So here's what the properly performed FIVC can do for you. In terms of risk, it can tell you what the confidence in the spirometry measurement should be. And the top row shows us what we see in about 55% of all measurements is that a properly performed FIVC, where the volumes match and the FIVC is inhaled rapidly, can leave you with confidence that any changes reflected in measurements that show that really are likely to represent changes in the patient's actual lung function and not a technical issue that's giving you lower than expected results at some measurements. The middle row shows submaximal inhalation error detected in 10%, and in different trials it averages about 8 to 10%. So it tells us that this technique is really, the results in submaximal inhalation error is really not uncommon, and it's probably more common than what's shown in that 10% prevalence figure. Because in the last row, that's where FIVCs are they're not being done properly. They're either inhaled too slowly, or the volume of the FIVC is too small to evaluate full inflation, or it's just not done at all. That's 35% of the data, and that's a bucket that's too large. Now, that doesn't mean the data in this row is bad. That 35% undoubtedly contains a very high percentage of measurements where the expiratory parameters are quite valid, and they really did start from full inflation. It's just that the site closed the measurement without demonstrating that, and so you, the sponsor, really don't know that. You don't have that confidence. And so, next slide. So what we do is provide sponsors with a variety of assessments and tools, starting with central overread, which we've covered. Outlier reports are basically looking at measurements that demonstrate big changes across serial measurements. Plausibility reports is looking via other techniques besides the large differences between measurements to look for implausible findings, things that just don't make sense. And one of the things that falls in this category is putting the wrong patient, testing the wrong patient under that subject ID. Sounds like it is uncommon, but it happens in virtually every clinical trial, sometimes multiple times. And finally, what we've spent the last few years developing is what's called a scientific spirometry quality report, which takes all of the factors above it, as well as information provided from custom dashboards we prepare for sponsors, and AI-generated tools that can help us look for all of the anomalous measurements, and then we do deep dives and report to the sponsor what we find with recommendations on what to do and say, what kind of conversations to have with the sites, with the operators, and what kind of measures might be needed, which includes possibly retraining. So that's a lot in a short period of time. And with that, I'm going to turn it over to Alon Gay, who's going to tell you about some innovative measures they're taking to address risk-based monitoring and cardiac safety. Alon?
Thank you, Kevin. Thank you so much for having us today. Next one, please. So today we're going to discuss a few tools that Cryo has been developing to help you with monitoring of your data. As you know, in the world of cardiac function, the two most common data that we capture are EKGs and blood pressure measurements. So we will review two of these aspects of this data with different tools that has been provided. We know that the way to capture an ECG has changed from the time I used to be in practice. From today, Holter ECGs are now common practice to also capture and extract ECGs. We want to ensure that the quality is optimal for this method of measurement. A second step that we take forward is trying to a little bit mimic what our colleagues are doing with spirometry, trying to identify if maybe subjects were misidentified during a trial, and trying to ensure that the data quality is being present. And finally, in the world of blood pressure, we will see a tool that we have been developing to ensure data quality as well. Next one, please. So let's start with ECG data quality. So in the next one, I will try to explain to you that when we collect ECG data using Holter, especially for QTc assessment, we are trying to now identify if the quality of the recording is as good as it should be. And to do that, we have used and developed an AI tool which is based on machine learning. Don't ask me too much about the technology. I know how it works. I will try to show, but this is highly sophisticated process, which I describe in the next slide. So basically what the tool is doing is that it divides the recording into epochs, which are small segments of the signal, and qualify them with a score. So each epoch will generate its own score, and you see this is a rolling method of assessment, and that will give you for each time point a total score based on each epoch, and that score will show on a visual scale as green is good and red is bad, so that quickly identifies good and bad quality sequences of signal that can help us identify where the pain points are. So we tested, and we show the results in the next slide. We tested this method of assessment with a bit more than 250,000 different epochs, and you see the results. So for the statisticians in the room, that will give you a statistics of 0.95, which is excellent prediction. So if we tell you basically that a true negative is a true negative, then you have a very good confidence. And the other way around, if you tell you that an epoch is good, you are pretty confident that it is good. So what does this mean? So next slide, we understand now that we have a tool which has the power to give you almost real-time feedback on the quality of the authors that are recording during your study, which means that we have now the capacity to tell you, even if you are, let's say, in a SAT study, where you want to ensure that you have good quality of data to make the necessary assessments, we can do that already from the lower dose cohorts. And even, I would say, maybe more important, for some of the clients, we also collect the data, but we don't process it straight away because that's what they want to do. And even then, we can assure them that data is there and that it is good quality, so that we can be able to process it further down the road should there be a need to do so. The second tool that we have been developed is a what we call a mirror participant detection tool. So we know that ECGs are almost like a fingerprint. So if everything stays equal, your ECG should not change over time, as I say, unless there is an external event that will change it. So we have conceptualized that in a mathematical setting that you can compare each ECG to each other within a subject or between subjects. And the tool is now capable of identifying if a patient is mislabeled, or I should say, an ECG is mislabeled to the wrong patient, or maybe it's also flag some suspicious or duplicate patients. Let me show you how this works. So in the next slide, we will see that we have created these embeddings. So the embedding is basically a distance between two ECGs, and you have to conceptualize that for the tool, the ECG is just a set of numbers that will go into a matrix, and then the tool will be able to assess the difference between these two distances, and that will give us something very graphical that we see on the next slide. That will give us a matrix. So in that matrix, you see a certain number of patients that are compared to each other. So you have to look only at the upper part of the matrix, the red part. So you see that the distance is visualized as a scale of color, and the darker the color, the more distance the patients, or if we are within one individual, the ECGs themselves, the darker the more distance, and the lighter the more similar they are. So of course you don't expect two different subjects to be so similar that they may raise attention. This is only for us the first step. So then we as cardiologists can deep dive into the data and see if these similarities are real or not. Of course, there are inferences like artifacts that can create false positive and false negatives. But like I said, this is a tool that is meant to grow on itself and learn as it goes. So this was for the ECG. Now let's move to blood pressure. So blood pressure monitoring, as we know, is also an important feature of safety monitoring during trials. So we know that our blood pressure varies every day, even from minutes to minutes. You sleep, you eat, you present something to a very nice audience, that will change your blood pressure. Eat cold also will do that. And there are also some procedural components. If the cuff that you are using to measure the blood pressure is too tight or too loose, or if there is a lack of consistency, so you put an ABPM on the right arm the first time and on the left arm the second time, that will create discrepancy in data. So what we did in the next slide, we developed a tool to monitor all this. And the tool, and I will not read you the whole slide, but you can see that the tool is built to detect these discrepancies and to detect variability that is not within a normal or expected, I should say expected range, more than normal range, because we also monitor blood pressure in patients, which can have higher blood pressure than normal individuals. And that allows us to give a periodic report in the studies where we monitor blood pressure to inform the sponsors about data quality issue, which may or may not be clustered in some sites or some patients. So then we can take together measures to increase data quality and to also increase credibility in trial results. Thank you. I will hand over back to Dr. Rudo for the conclusion. Thank you.
Super. Thanks, Lon. So some really interesting applications there in blood pressure and ECG data as well. I appreciate you taking us through them. One slide here just to close us out and transition into a Q&A session. Key takeaways are listed here, but just to quickly summarize, we heard today a really compelling set of examples for the impact that novel risk-based monitoring methods can have in improving clinical trial data quality, benefits for efficiency and cost, but also for data accuracy and trustworthiness. The advanced analytic strategies Stacy took us through in ECOA data were quite impactful, benefits for participant level data, site level data, and also identifying protocol level risks as well. In spirometry, you heard from Kevin about the novel applications for detecting anomalies in that unique and challenging data type, spirometry. The key there is that opportunities for site education could be readily identified to improve the data quality during study conduct, in addition to the ability to identify any potential deliberate site misconduct, which may be ongoing. And in cardiac, we heard about novel applications for continuous ECG, for standard ECG, clinic blood pressure as well. Again, retraining opportunities, but also identification of potential misconduct as well. So let's jump to some Q&A in the remaining time. Stacy, Kevin, Alon, I'll invite you to come on screen here with me. The first one, let's see, this first one here for Stacy that has come in. Considering that there are varying types of ECOA, so patient-reported, clinician-reported performance outcomes, are there differences in how teams should be approaching RBM monitoring based on that type of ECOA?
Sure. Yeah, great question. The short answer is yes. We really want to try to avoid a one-size-fits-all approach to monitoring and really make sure that we're taking a look at the unique aspects of each COA measure alongside the protocol, the specific indication, visit schedule, and any other sort of COA measures that are there. So it really does require a customized risk-based monitoring plan to consider all of those factors, led by teams who have some expertise in both, the COA measures themselves, the protocol, population, and then also a team who can also support the actioning, so what you're going to do about it. So some things to consider too are based on the type of findings that you might identify with risk-based monitoring, really trying to take a custom approach on how you're going to action it, or if you're going to action it versus monitoring it. So I think all of those things come into play. It is really important to look at the type of ECOA measure when you're developing your monitoring plan.
Okay, great. Thanks, Stacy. Kevin, this one's for you. I like this one actually. It says, it seems like you're suggesting that a higher percentage of spirometry data should be considered unacceptable than what already is when the more standard ATS-ERS standards are applied. Why is this a good thing?
Well, it is now part of the standards that if you demonstrate submaximal inhalation error, you really haven't satisfied the first tenet of quality spirometry, that the forced vital capacity was always intended to start from full inflation. And therefore, when you get a measurement that starts significantly below full inflation, even though it looks perfect otherwise, it really doesn't represent what the patient's condition is at that point in time. In other words, what you're doing is you're introducing noise. You're saying, oh, we don't want to call too much data unacceptable, so it looks good otherwise, let's just accept it. That's the moving in the opposite direction of where we want to go. So there is a pain to calling measurements unacceptable that people used to think was acceptable. But with education, you can really stress the importance of full inflation and start reducing the noise and all the attendant benefits of that.
Yeah, that's great. Yeah, net benefit certainly for overall data quality, even if some challenges along the way there. So thanks, that's helpful. Alon, this one is directed at you about that mirror patient tool that you described. So we know that ECGs can change within a given participant over time because of clinical events, such as a new arrhythmia or a recent MI, so that's myocardial infarction. Would this not create a false positive finding in your tool?
Thank you, Todd. That's a great question. So yes, indeed, this is one of the challenges that we are facing today with the tool, and that will indeed create a false positive. And this is why I should say on top of the tool, we have cardiologist oversight to check these findings and confirm if they are real or not. Another typical situation, like I explained in my presentation, are artifacts. But this is also the beauty of the tool is that this is an AI that is learning. So the more we will use it, the more it will learn, and it will learn from these mistakes, I should say. And hopefully next time we'll be able to discriminate better.
Okay, great. Appreciate that. Yeah, and if anyone's attended some of our prior webinars where we've talked about AI tools, that human oversight piece is key, and obviously each tool needing its own sort of bespoke level of human oversight, and in this case, as Alon mentioned, cardiologist oversight. So, Stacy, I'm going to come back to you on this next one. It's a good challenging question, actually. You mentioned actionable insights resulting from RBM of ECOA data. Can you provide some specific examples of actions that may result from the RBM applications?
Yeah, sure. Great question. Yeah, so I mean, the reality is when we put these plans together, or when sponsor teams, monitoring teams are putting these plans together, you're really trying to think about like, wow, I have all of this data. It's all in one place, which is great, but there's so much. What am I going to do with it? What am I going to do about it? And I do think it's important to consider, well, what are you going to do about it? What actions you're going to take as you're developing it? And again, just like your plan itself is going to be customized to your protocol, phase of study, I mean, all the unique characteristics of your study, the actions you take might vary as well. So some examples I can provide. So protocol retraining, right? So oftentimes the data emerging might show that there is perhaps misperception, misunderstanding of protocol requirements, when scales should be administered, the expectations of rater consistency, things like that. Other times we might flag that there are concerns with understanding of the population, or maybe how the scale was administered, how the scale is expected to be administered, or even scored. And so that's where we can do some retraining. We can, there's a number of methods of retraining or remediation, providing direct feedback to sites and trying to work with them to get recalibrated. So just a few different actions, but we try to work together, really kind of partner. It's important that your monitoring team is partnering with sites and sponsors as kind of a solid partnership among the groups to make sure whatever actions you put in place are going to be useful and meaningful for the outcome of your study.
Okay, super. Thanks, Stacy. Sonia, I think I'm going to invite you back on screen here to close us out. Thanks to all the presenters for your contributions today.
Okay, well, thank you very much for answering all those questions. We have reached the end of the Q&A portion of the webinar. And remember, if we couldn't attend to your questions, and there's a lot of them out there, the team at Clario will follow up with you after this presentation. Now, if you have any further questions, direct them to the email address on your screen, science@clario.com. Thank you, everyone, for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve our webinars. Additionally, I have shared a link to view the recording of today's event in the chat box, which you can share also with your colleagues once they register for the recording here as well. And for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to such features as viewing the recording and more. Now, please join us in thanking our speakers for their time here today. So thank you, everyone. And thank you, Todd, for being my co-moderator for today's event. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Bye, Stacy. Bye, Alon. Bye, Kevin. Bye, Todd. Bye, everyone.